So our project is generating novel variation in soybean microRNAs to improve drought tolerance. We're just in the initial stages of this project now. I'm in the second year of my PhD program in Dr. Ma's lab. And so this is a project that should continue through the end of my time at Purdue here. So just briefly, as many of you know, soybean is very important. It's the most widely grown legume in the world. And I just want to draw your attention to the fact here that world soybean production is increasing, and that's because demand is increasing. And the map on the right here just shows some major centers of production for soybean. So the United States, Brazil, Argentina, along with China and India here in Asia. This is an interesting chart that shows soybean uses that was put out by the North Carolina State Extension. And the reason I put this on here is just to draw your attention to the large amount of soybean that goes into animal feed. And the reason that that's important is because, as well as these other uses, is because the demand for these products as the global population grows, and especially as the global middle class grows, is going to increase. And so we need to maintain soybean production to meet these demands. And hopefully this proposed project will be a contributor to that. So abiotic stresses are major limiting factors to crop production, including, of course, soybeans. Abiotic stresses include cold, wind, high salinity, and here we're going to focus on drought. So drought's a major problem. It can reduce soybean yields by more than 50%. Oftentimes, the severity of the loss relating to drought has to do with the timing of it. Different stages of soybean growth are more sensitive, especially the early vegetative stages soon after planting, when we are trying to establish the stand in the field. Also, during flowering and seed fill have been identified as being really sensitive times. If we have drought during that period of soybean development, you see pretty substantial losses depending on the timing of the drought. So drought stress results in morphological, biochemical, and physiological changes within the plant. The biggest problem is the loss of water potential gradients that are necessary for plant growth in terms of transporting nutrients and other things into the plant and across membranes within the plant. Many changes in response to drought stress are mediated by abscisic acid, ABA, and genes from a variety of families show expression change in response to drought. So this is a figure from Shinozaki in 2007 that I think sort of illustrates this well. We have functional genes that are responding directly that are affecting the response, like detoxification enzymes or water channels and transporters, as well as regulatory genes like transcription factors, protein kinases involved in signaling pathways, or ABA biosynthesis. And in addition to protein coding genes, we also see differential expression and response from regulatory RNAs like microRNAs. One other problem I wanted to note that's more specific to legumes like soybean is drought causes a reduction in nodulation and symbiotic nitrogen fixation, which is very important, of course, for soybean growth. So breeders have used a wide variety of strategies to improve drought tolerance. These have included larger and deeper or otherwise modified root systems to try and increase water uptake, modification of stomatal density or aperture to decrease transpiration, to decrease water loss, as well as increased biosynthesis of osmoprotectants to stabilize those membrane gradients I mentioned, or to scavenge reactive oxygen species, which are harmful molecules that are often produced during drought stress or other abiotic stresses. Modification of ABA signaling pathways, and finally, as I mentioned before, with legumes, maintaining symbiotic nitrogen fixation. So drought tolerance is a very quantitative trait. It's been under selection pressure both in the wild and by breeders for a long time, and so there are a lot of loci that have small effects, which poses a challenge. And one of the ways that we've tried to get around that challenge is through gene editing. And CRISPR-Cas9, as a lot of you now know, is a useful tool that allows us to do that. And an emerging area of interest for crop improvement is using gene editing techniques to modify microRNA genes. And so that's what the focus of this project is going to be about. So you might wonder, well, what is a microRNA? MicroRNAs are 20 to 24 nucleotide small RNAs. They're transcribed from microRNA genes here. These transcripts contain what are called hairpin loop structures, which you can see in this figure. And they're double-stranded because we have complementary base pairing that forms these loops. And the cell doesn't like to have double-stranded RNAs there. So these endonucleases called DCL1 will come and cleave that into this small double-stranded fragment, which gets exported out of the nucleus into the cytoplasm. It's unwound by some helicases so the two strands are split apart. And then one strand gets incorporated into a complex with an AGO protein, forming what's called an RNA-induced silencing complex, or RISC, that can be used to regulate gene expression. I know that's a lot of information really quick. It's maybe a little confusing, but the key takeaway is that microRNAs are these small regulatory RNAs that can play a big role in regulating gene expression. So then you might say, how do they regulate gene expression? And in three general ways. The first way is through directed cleavage of target mRNA transcripts. So this figure from Rogers in 2013, I think, does a nice job of illustrating the different ways in which microRNAs excuse me, can regulate gene expression. And so the first way is through directed cleavage of target messenger RNA transcripts. So you can see down here, the microRNA is acting as a guide for this RNA-induced silencing complex to target this messenger RNA, and it's going to cut it to delete the function of it. Another way is through translational inhibition, so preventing the transcript from being turned into a protein. And then finally, some studies have also shown examples of microRNAs guiding methylation to regulate at the transcriptional level. So microRNAs have also been studied for their role in drought stress. Firstly, microRNAs have been very well characterized when it comes to development. And so differences in development of roots in particular and other plant structures can affect drought response, of course. And microRNA 160 and microRNA 390 are just two examples of that. What we want to focus more on, though, in this project is microRNAs which show differential expression under drought stress. And this has been identified in many plant species now, including soybean, maize, rice, Arabidopsis, and a few others. And in Arabidopsis, knockouts of microRNA 169A and microRNA 827A using CRISPR-Cas9 improved drought tolerance. So this is a viable strategy. A lot of the details remain to be worked out, whether we can use this in a way that not only improves these specific traits, but also ends up benefiting us from a yield perspective. There's still a lot of work to be done. So our general strategy and outline of our objectives here, we want to firstly identify and evaluate microRNAs or their targets, which could be useful for editing to improve soybean drought tolerance. We then want to modify those selected microRNA genes or their targets in an elite cultivar with targeted deletions using the CRISPR-Cas9 system. We then will evaluate the response of these edited lines to drought stress, as well as high salt stress, because that's a very related abiotic stress, and many of the genes which affect those two pathways are the same. And we want to also characterize any related phenotypic changes, differences in plant growth, response to nodulation, and other phenotypic, maybe pleiotropic changes that we get from changing these microRNAs. And then finally, we want to select transgene-free progeny to evaluate the effects on yield in a field setting. So the first step, as I said, is to identify candidates for editing. And there is publicly available small RNA sequencing data, messenger RNA sequencing data, and degradome data, which we can use to identify microRNAs and their targets, which show differential expression in response to low water stress. And once we have those, which we did, we can take a look at the current literature as well as the gene annotations and other information that we have, plus results from our own experiments to get an idea of these candidates. So we looked at both microRNAs and their target genes that they regulate to be evaluated as candidates for editing. And certainly we're not limited to this, but ideal microRNA candidates should be abundant. They should show a decrease in expression after exposure to drought stress, and we should see their targets increase after exposure to drought stress. because this would indicate that these microRNAs are acting as negative regulators of drought tolerance. So I want to make a quick note about CRISPR-Cas9. It's a very versatile tool. Some of you probably have used it in your own research, but it allows us to make targeted deletions of genes called knockouts, right, where we are targeting the coding sequence of the gene in a way that we completely eliminate that gene's function. Another option is what are called knockdowns, or targeted deletions in the promoter regions in an effort to modify the expression level. And if we have multiple targets within the promoter region, because of the editing efficiency of CRISPR-Cas9 is not perfect. Actually, it can be somewhat low. We should get different deletions and different combinations of deletions within those regions that can allow us to generate quantitative variation in the expression of our target gene. And that's a strategy that's been used successfully in some other crops, such as tomato. So now what I want to do is talk about some of the candidate genes that we've selected in our preliminary analysis. The first of those is microRNA 166N. And actually what got our lab interested in this project of looking at microRNAs and drought stress was that our lab had done a study of the MADS box transcription factor DT2, which has a dominant gain of function mutation that regulates stem architecture. But in addition to that, it also was found to reduce tolerance to drought. And so part of the way it does this is by it targets and reduces the expression of this microRNA 166N. And so microRNA 166 in rice was found to target OSHB4, which is an HD-ZIP3 family transcription factor, which regulates cell wall and vasculature development. And in rice, it was found that knocking out this microRNA improved drought tolerance through modifications of the leaf structure. And so in the data, we looked at several microRNA 166 family genes, including 166N, were downregulated by drought stress. And they had target HD-ZIP3 family transcription factors, which were upregulated, including GMHB15A, which was identified in our lab's previous study. And so the hypothesis for this microRNA 166N is that if we can knock out microRNA 166N, we could also see improvements in drought tolerance in soybean. The next one is microRNA 4996. In the data we have available, we found that at 12 hours post polyethylene glycol treatment, and polyethylene glycol acts as a mimic of drought stress, its expression is reduced by half, and its predicted target has a twofold increase in expression. And its target, one of its targets, is this gene on the 6th chromosome, which is a tyrosinase involved in oxidation reduction, which, as I mentioned in the introduction, is an important component of stress response. And so what this shows here is just the top is the microRNA showing its complementarity to this target gene, which was shown in the degradome data. So the hypothesis with this microRNA, again, is that if we knock it out, we can improve drought tolerance through increase in the ability of oxidation reduction through this target gene, not being downregulated by it. The next ones are the microRNA 1508 family, 1508A and 1508C. And actually, microRNA 1508 is one of the few microRNAs that has been characterized for its role in drought stress. It was found that overexpressing microRNA 1508A reduced drought survival while also increasing cold tolerance. So again, that illustrates the pleiotropic effects and the multiple pathways that these microRNAs can be involved in. microRNA 1508C has not been characterized, but in the data we looked at, both of these microRNAs showed significant, decrease in their expression during drought stress, while their targets, some of which overlap, increase in expression. And so for this one, this will be an example of where I think that using the knockdown strategy of targeting the promoters would be an effective strategy. So the hypothesis is that if we construct guide RNAs within a Cas9 construct that can target both of these promoter regions of these genes, we should get some variations in some different combinations of expression levels for both of them. which should allow us to find sort of an ideal level of expression where we can maybe increase drought tolerance while not affecting that cold tolerance that the other group mentioned. And then finally, microRNA 1507B. This again was downregulated by drought treatment in the roots specifically. Its predicted targets, or its targets, not predicted, but confirmed targets by degradome were NBS, LRR, or leucine-rich repeat genes that are involved in defense of against pathogens. And a previous study also showed that this microRNA was found to be upregulated in response to rhizobium inoculation, which indicated that it may play a role in suppressing plant defense responses to allow for symbiotic nodulation. And since we know that drought, which you can see here in this picture, reduces nodulation, the hypothesis is that perhaps the downregulation of this microRNA 1507B is allowing for an increase in the expression of these NBS LRR defense response genes, and therefore reducing the ability of these symbiotic relationships to be formed in the nodules. And so the hypothesis for this one is that reducing the expression of these targets, these NBS LRR genes, not knocking them out completely because they're important for pathogen response, but reducing their expression could help to maintain nodulation during drought stress, which would be a very, very nice thing for soybean to maintain its nutrients. So the outline of this project, or sort of our timeline here. Right now we're working on our preliminary data and our selection of these candidates. The next step will be confirmation of these microRNAs drought responses. Following that, you'll need to design our Cas9 vector constructs that will lead to soybean transformation and tissue culture. We'll phenotype T1 edited plants, which are homozygous for these edits. And then finally, in the long term, in the following summer, we'll do field evaluations of these edited plants. So I want to quickly just give a brief overview of the methods that I haven't touched on yet. So for our verification of expression change for these target candidates for editing, we're going to use polyethylene glycol, which, as I said, mentions drought stress at the V1 stage in a protocol modified from Joe et al. In 2020. We'll take leaf and root samples at 0, 6, 12, and 24 hours post-exposure to polyethylene glycol. And the expression of these microRNAs will be measured using stem loop RT-qPCR. And all stem loop RT-qPCR means is that when we do the reverse transcription step, we have special primers that allow us to focus on these microRNAs. Our lab now has a lot of experience in doing transformation and tissue culture. This is often challenging in soybean, but both myself and other members of my lab have now become pretty good at this, so we feel confident that we should have no issues at this stage. And this picture on the left here just shows some transgenic plants in the rooting media stage of tissue culture. And then finally, we have to evaluate abiotic stress after we make these modifications. So in the greenhouse, we're going to do drought and salinity assays of homozygous edited T1 plants, or we may not have enough T1 plants, so that the backup would be to use the T2 plants, the next generation. To be carried out in the greenhouse in a protocol modified from our lab's paper in 2019. Salinity tolerance will also be assay will also be carried out in the greenhouse using a protocol modified from Lee in 2004. And then finally, we will do we'll find transgene-free edited lines and controls to plant in the field at acre for field trials. Because ultimately our goal is to have this be an applied project where we can really make a difference in addition to understanding these pathways. So in conclusion, drought is a major abiotic stress limiting soybean production. MicroRNAs are important regulatory molecules that can be manipulated for crop improvement. We've identified several microRNAs and their targets, which may play important roles in drought tolerance in soybean, and we will modify them using the CRISPR-Cas9 system. Ultimately, this will allow us to characterize the roles of these microRNAs in drought response in soybean, and will facilitate the development of lines which can maintain yield under drought conditions. We're right at the beginning of this project. There's still a lot to do, but I appreciate everyone coming and listening to this presentation today. I want to thank my major advisor, Dr. Ma, along with my committee members, Dr. Teinstra, Dr. Mickelbart, and Dr. Rainey. I'd like to thank all my lab members who were always very helpful, the United Soybean Board, which is partially responsible for the funding of this project, and also thank you to Richard and to Charlotte for helping to host this, and to Charlotte for making the flyers that everyone saw. So I've got my citations in the next few slides, but I'll open it up to questions if anybody has any.
Okay, questions for Chance. If you have a question, unmute, let them know who is speaking, and then ask your question.
Chance, this is Mohsen.
Oh, hello, Mohsen. How are you?
Good. How about you?
Doing okay.
Great. Great presentation. Thank you.
Thank you.
My question is a little bit general. I want you to define what is drought. If you are a plant, how do you see the drought? And then what is drought tolerance? What are the modes that plant uses for combating against the drought? And a little bit of those terminologies, if you've encountered those, because that can make a good introduction to your thesis.
Okay, so I would define drought just in general as the plant not having adequate moisture to maintain its normal standards of living and growing. I think that's probably the best way to define it simply. In terms of drought tolerance, I think that would be best defined as the ability of the plant to maintain its normal growth patterns in response to that stress. And some of the ways it does that go back, I think here this is talking about targets for improvement, but really these are also would be related to the natural ways in which the plant responds. So especially through ABA signaling, we see increased stomatal closure to try and limit that transpiration, to limit water escape. We see morphology change where in response to drought, plants tend to increase the mass of their roots relative to shoot growth. We also see synthesis, again, of these osmoprotectants that stabilize these gradients, as well as molecules that can scavenge some of those reactive oxygen species. So I would say, so those are some of the general ways. Does that answer your question?
Yeah, that is good. Yes, there are two papers. I will send it to you. One is from Levitt in 1985, and then there is one from Bloom. He is a pioneering drought response person, and there are terms in those that it is going to be very useful for people doing abiotic stress tolerance. Terms such as drought avoidance, drought tolerance, things like that. So I will save my other question and I let other people to ask. And if there is no question, then I will continue.
I appreciate you sending me those papers, and
yeah, sure.
And you're right. I do need to maybe incorporate some of that terminology.
Sure. I'll send them to you.
Oh, thank you.
Yeah.
Does anybody else have a question they'd like to ask?
Maybe me again?
Go ahead.
All right. So this question is a little bit more targeted toward the concept you're researching. So we know that let's say different soybean varieties, there is natural variation in the expression levels of the genes, of the protein coding genes, correct?
Yes.
Like in a baseline expression, the soybean varieties may differ from one another. So this question that I have is going to ask you, do you think in microRNA, do we see a baseline differences in their expression? Like are there varieties that, let's say these microRNA number 169A. The baseline expression of it is different, so you can go and use that in a breeding program rather than just going through the hardship of doing the genome editing. So what I'm saying is, why not natural alleles? And why bothering making the new variations if there are natural alleles?
There is natural variation in these expressions. Actually, that's something that, and there have been studies that have shown that, but a lot of work remains to be done to identify that natural variation, I think. The reason we would want to use this DNA editing as opposed to the natural variation is that it allows us, one, to solely focus on that target. We feel comfortable that we can carry out these Cas9 edits in a pretty efficient way in our lab without the need to do a lot of back crossing to, it could be that a line which has better expression of one of these microRNAs we're interested, or reduced expression. It could be that there's some negative traits in that as well. So we would need to do a lot of back crossing to get rid of that linkage drag that might be associated with that. And so because we feel confident in our ability to do this, this is maybe a faster way to just focus on that directly, as opposed to having to deal with all the other issues that might come up with doing traditional breeding.
Thank you. Thank you very much.
And also, of course, for the ones we want to actually knock out, that level of variation probably doesn't exist in nature.
I see. Good point.
That's a good question. Thank you. Does anybody else have a question?
Hello, Chance. This is Charlotte.
Hey, Charlotte.
And I'm going to kind of build off of some of these questions about drought and terminology. And sorry if I missed this earlier in your presentation, my internet's been going in and out, but I was curious how maybe you would be testing some of these, I guess, based on how you were defining drought as the plant not receiving enough water and that impacting its growth functions. If you were testing out these different strains or genes in a setting, how would are you thinking of, in your definition of drought, is it restricting water an equal amount throughout the whole period of growth, or is it coming at different times during growth? I'm curious if you've thought about that in some of your work.
So what we're going to focus on initially is the early vegetative stages. So when we do our initial drought assays, where we just want to confirm the expression of these microRNAs in response to drought stress that we found from both our own data and some other publicly available data, will be done in the greenhouse using polyethylene glycol. So polyethylene glycol can mimic drought stress. It activates the drought stress response pathways. Once we have our edited lines, we will first test them in a greenhouse setting. So that'll be in a media that doesn't hold water well, so that we can precisely control the amount they're getting every day. So what we'll do is give them normal water up to a certain stage, and then we will withhold water or reduce the amount of water for a certain length of time. And that still probably needs to be determined a little bit, but it'll be based on some experiments our lab has done in the past with drought stress. But primarily it's going to focus on recovery. So at some point we'll, after a certain amount of time, we will go back to a normal water regimen, because that's what we'd expect to see in field conditions, right? I mean, sometimes you have a whole season drought, but normally what happens is you have a certain period where there's not enough rain, and then the rain comes back. So the ability of the plants to, once they have water, to come back and be okay and grow, that's probably the main focus. Does that answer your question?
Yeah, thank you for helping clarify that for me. I think a little bit of that comes from different uses of drought in our terminology. Since I'm a hydrologist, how I use the drought may be a little different than how you're using it. So that helps clarify it for me. So thank you very much.
Okay, any other questions?
One more.
Go ahead.
All right. So you want, let's say you want to work on microRNA 169A, for example, for the sake of example, and you want to knock it down because you believe that under drought, this goes up and the expression of the target gene will go down. So you want to knock the microRNA down in order to get more expression of the target gene, correct?
Yes.
Okay. So what does this microRNA do when there is no drought?
So that's one of the things that we looked at. So one of the, for example, one of the microRNAs that we saw that showed a big drought response in terms of its expression was microRNA 156. And 156 is really involved in plant development, especially through controlling juvenile to adult stage phase transitions. And so, for example, that one we excluded as a target because we said if we mess with this, there's so many things this is involved in that we know, okay, this is not going to be a smart choice because the pleiotropic effects are going to be probably very negative. So all of the ones we looked at their target genes. For example,
the one with oxidation reduction.
Yeah, so for example, this one is reducing the expression of this tyrosinase, which is involved in oxidation reduction. And it doesn't have many other targets, actually, at least according to the degradome data. And also, just even with the annotation, the higher number means that it's probably more unique to soybean or to legumes, and so it's probably less conserved a little bit. And so that was one of the things we carefully considered. I went through and looked at each one of the targets of these genes. I went and looked at their expression. I went and looked to see if any other information existed about these to make sure that we weren't targeting one that was likely to have some really negative effects. And that's part of the reason why we want to do field trials. There's been a few studies in Arabidopsis and rice and maybe one or two in soybean where they've modified microRNAs and they've said, okay, we show that it improves this trait. But they didn't go on further and say, okay, how does this actually affect the yield of the plant? What are the other effects of this? You know, how does this affect the yield when we don't see a stress condition? And so that's why I want to do field trials at the end of this to really evaluate the total phenotypic change and role of these microRNAs.
Good points, Chance. Thank you.
Chance, are you okay?
I'm good, yeah.
So you did a good job. I don't have a question.
Okay, are there any other questions?
And Mohsen asked a lot of great questions.
Oh, he does, yes. Well, Richard, I guess if there's no other questions, we can wrap it up. Thank you again, everyone, for taking the time to come listen. And Richard, I guess, probably knows who goes next.
Yeah. So Chance, thank you. Thank you, Mohsen, for the questions. Again, I still think that Chance is getting a little bit easier than Seth did a couple weeks ago, but you know, that's all right. So we'll have 3 more presentations this semester. The first of which is next week, and Charlotte. It's going to be Charlotte Lee presenting, and she's going to be presenting an update from the Purdue Hydrologic Impacts Group. So all the work that's being done within that group. And then after that, we will have 2 more proposal presentations, one from Christina Schilling and the other from, to be honest, I can't remember who it is, but I believe it's on digital soil mapping. Those will be at the same times on Fridays at 3 o'clock, other than Thanksgiving week. Obviously, there will be nothing there. With that, I just want to say, everybody have a nice weekend. thank you for participating, and hope to see you next week. Nice job, Chance.
Thank you, everyone.
Good job, Chance.
Great job.